Hello learners, welcome back to my YouTube channels. After a very long time, I'm going to tell you or explain you about the very interesting technique which you all must have to know. And I think many of you have heard about this. So before wasting any time, just let's get started. So first, hybridoma technology. The Hybridoma technology is very important immunological technique or applications of this immunology because it has the ecological values so many, and as well as the tremendous medical and the treatment values. So we'll discuss it later on. So first of all, the as we all know that polyclonal antibodies are there and monoclonal antibodies are there. So polyclonal means they are not specific to only one kind of antigens. They can bind with many types of antigens. This is said to be a polyclonal antibodies because our human body can affected by so many, or can in every second we are exposed to different types of antigens. Very Many types of foreign particles are entering in our body, so immune response is generated, and this in response to these antigens, there are so many immunological cells, immunocytes like lymphocytes, B lymphocytes are triggered to produce this antibody. This antibody are polyclonal antibodies generally. But if we want the monoclonal antibody, what is the importance of this is it is highly specific to only one kind of antigens. So our idea is to produce this monoclonal antibody by this technique called as hybridoma technology. How can we do this? Let's see. So first of all, there are in this idea we are fusing the we are hybridizing the two types of cells. First of all, the myeloma cells, which are the cancerous cells, and the second is, so in this technique, our idea is to fuse the two types of cells. The first is myeloma cells, and the second is the B cells, or normal lymphocyte cells. Myeloma cells is a cancerous cells which has ability to produce immortality. Immortality, it produce itself. It has no limitations to produce in the specific number. It can generate so many numbers of that, and we have to take this characteristic of these cells, and we use these cells with the normal B lymphocytes. B lymphocytes are the ability to produce the antibody. Okay, so these B lymphocytes and the myeloma cells are fused in such a way to get the hybridomal cells. These hybridomal cells has two characteristics. Now you all know the immortality and the antibody producing. Now what our aim is to produce the monoclonal antibody. So how and when, let's see. First of all, there is some protocols in this hybridoma technology. In the first step, we are injecting the lab rat with the specific antigen that we want to from on which the molecular antibody uh, to work on. Okay, to work on or to interact with. So this specific antigen injections is injected in that particular lab rat, and so that in the rat there will be immune response will be generated, and the antibody will be produced, or B cells will be activated. These activated B cells of the spleen of the rat is isolated from that rat, and this B cells is in other sites. There will be myeloma cells which are being isolated from that mouse. Myelomal B cells means this rat is somewhat infected with that cancer. Okay, so this is how we isolated the two types of cells, B cells and myeloma cells, and now this is third step is to fuse both the cells. Okay, how? So there will be a gel, we call it as I forgot the name, but yes, Okay, so we fuse or hybridoma these two cells. Okay, and as soon as the hybridoma cells are produced, there will be chances of myeloma cells are also fused with each other. There will be chance of the B cells and B cells also fuse with each other, and some cells do not even fuse. Okay, so how we get only the hybridoma cell from this cell culture? So what we are doing here is we are doing this selection process. This selection process we can do because of the characteristics of this word the cells. Let me clear one thing here. The B cell has an gene. The B cell has a gene. First of all, this selection is done in the media HAT. Okay, HAT media. This is a HAT media. Again, I forgot the name of the. So the selection process is done in the media called the HAT. The full form of HAT is the hypoxanthine aminoprotein thymine. Hypoxanthine aminoprotein thymine. So in this media, there will be enzymes which is able to hydrolyze these the hypoxanthine to form the nucleotides. These enzymes are generally produced by the B cells because the B cells has the genes the HPRT1, so which codes for the enzyme the hypoxanthine guanine phosphor phosphorybosylase transferase. Hypoxanthine guanine phosphorybosylase transferase. Okay, these enzymes make the hydrolysis of these hypoxanthine to form the nucleotides. Okay, only these enzymes can do this, and only the B cells, these are the cells which possess these enzymes. So these cells which are fused with B cells will only grow on this HT media, and the rest one will be die. Now what about the B cells and B cells myeloid cells? Because B and B fused cells cannot grow immortality, they will be eventually die. So we only get the B and myeloid cells, hybridized cells. Okay, this is how the selection process is done here, and which is very important process of this technology, and great care should be taken during the this production of these hybridoma cells, and from which the monoclonal antibody. Now as soon as we get the abdominal cells, we are now allowed to grow, we are allowed them to grow, because we want these cells in very good quantity also. So we grow these cells in the media, or we can say we culture them in a tissue culture. Okay, in a tissue culture we inoculate these cells and like some animals, okay, or rabbit or rat, and allowed their productions, their proliferations, and as soon as we get their proliferated the cells, we allowed to we specifically inoculate the antigens that we are going to work on. Okay, so these antigens injections is injection are injected on that animals, and these hybridized cells are triggered and produce the antibody, which is now the monoclonal antibody. So this is the beauty of this technique, and as soon as we get this monoclonal antibody, these monoclonal antibody are the inoculated or isolated from that animals, and stored in they are stored for further analysis and uses also. So applications of monoclonal antibodies are tremendous. They will be diagnostic mechanisms like HIV diagnosis, also important in that, and their applications are very very important for us, as well as most of these companies, the pharmaceutical companies, are dependent on this hybridoma technology process. And so there will be great great price money will be there on their productions, but great with great price the great care should be done. The extremely professional work it is. And as soon as we are we get the perfecties or expertise on this making this antibody, believe me, you can get so many money from this, and companies are there to produce this monoclonal antibody. Okay, these monoclonal antibodies are very important to kill the cancer cells. Yes, you have heard the right thing, the cancer cells. We can kill these cells by the use of this antibody. How? Let me clear you. So just suppose that this antibody can this monoclonal antibody is specific to certain antigens, so it can targetly bind to that cancer cells. And this antibody is coupled with the drug that usually the radioisotopes. Okay, this coupled antibody targetly bind to that cells, and they will be killed, and without any side effects to the other healthier cells. Okay, this is how the killing of the cancer cells also done, means a treatment. And also there will be the best example of this use of this monoclonal antibody is the pregnancy test. Yes, pregnancy test. Yeah, we are doing here is depending on this monoclonal antibody productions. Okay, so I hope you are now clear about this monoclonal antibody, or we can say the hybridoma technology and their importance also. So thank you very much. Any questions you can ask me in the comment sections. So if you like my video, just like and subscribe to my YouTube channel. Thank you.